This is Professor Scher. This is a little lecture about some parts of proteins. You will need to supplement this information with the excellent exercises on mastering biology, the videos posted online, and of course your textbook. But let's get started talking about the basics of proteins. So why do we care about proteins? So most of a cell, if you recall, is water. But when we take out the water, what we call the dry mass of the cell, 50% of that is protein, which means that they are significant as far as the structural and functional levels of cells go. Proteins have a lot of different functions. So here are something like 7 different functions or 8 different functions proteins have. There are links online that go over the different types of proteins and their functions, so you'll want to look at that. If we look at them briefly, there's this nice table in your book. You can see enzymatic proteins are going to be very important, so you definitely want to understand those. We also have defensive proteins, so there's some examples there. Storage, transport. We'll talk a lot about transport proteins in Unit 2. Hormonal proteins, receptors, also part of Unit 2 and Unit 3. Contractile and motor proteins, part of Unit 2, and structural proteins. Now all these, of course, are involved in all the units because that's how cells work, and cells, of course, are the basic units of living organisms. Some people get a little confused between what a protein is and what a polypeptide is. So a polypeptide is a series of amino acids that are covalently bonded together. Each of these circles is an amino acid here, which we'll talk about that in a minute. And of course, these are covalent bonds. So this chain, that's the polypeptide. The protein is this chain folded up into some specific shape and then possibly combined with other folded chains in order to make a molecule that actually has a biological function, whether it's building structures or catalyzing reactions, one of those functions that we just saw. So there's a difference between polypeptide and protein. And polypeptides are the polymers of proteins. If we look at the monomers of proteins, those are going to be what we call amino acids. And here's the structure of an amino acid. You can see it has 4 main components. So the amino part of the name comes from the amino group that's on the amino acid. So remember, that's a nitrogen bound to 2 hydrogens. The acid part of the name comes from the carboxyl group, because remember that hydrogen can fall off and increase the acidity of the solution, which means that acts as an acid. So amino acid attached to a carbon, which is attached to a hydrogen. And then we have this side chain, or R group. So that group is going to be different in different amino acids. So this whole molecule, that's the amino acid, the monomer. There's 20 different types. It doesn't have to be drawn in this orientation. For example, here we can see here's the amino group. Box that for you. So here's the amino group in this one. Here's the carboxyl group, your central carbon, your hydrogen, and then this group here would be your R group, your side chain. So you see we've just taken this guy and turned it on its side. So basically what you're looking for is you're looking for a carbon, carbon, nitrogen in a row. That's going to tell you that an amino acid is there. You're looking for carboxyl groups and amino groups as well. If we look at the 20 amino acids, we can kind of group them into 3 main categories. These 2 have subcategories within it. And basically that's based off of their side chains. So you can see the backbone, we call this the backbone, is the same in every single amino acid. Amino group attached to a carbon, attached to a hydrogen, which is also attached to a carboxyl. The side chains are what give the amino acids different characteristics, which is what makes different proteins behave in different ways. So here's a group that have nonpolar side chains. You can see that we have lots of carbons and hydrogens, which is not surprising. These rings are also made of carbon and hydrogen, because remember those are nonpolar bonds, which means they are nonpolar side chains, and so these are not going to interact with water very well. They can't hydrogen bond with water, so they are hydrophobic. So if you had any of these amino acids, you're expecting them to fold to the inside of a protein because they'll hide from water, in a sense, because they don't hydrogen bond with it. Each amino acid has a name. It also has a 3-letter abbreviation and a single-letter abbreviation as well. You don't need to memorize any of the amino acids. But what you would want to be able to do is if I showed you something like isoleucine, you could figure out that it's nonpolar and figure out what does that mean. That means it's going to be in the inside of the protein. That means it's going to be not interacting with polar or charged molecules in the protein and that kind of thing. These are the polar side chains. Unlike the nonpolar, these guys are going to contain lots of hydroxyl groups, some carbonyl groups, and then we have even a sulfur or sulfhydryl group here. So these are going to be important for, and they'll interact with water. They hydrogen bond with water, which means they'll be on the outside of the protein. Or sometimes if we have a protein like in the cell membrane, and we want polar or charged particles to be able to travel through it, the little tunnel through which they travel is going to be lined with these polar amino acids so that when the charged particle goes through, it interacts with them okay and can travel through the membrane. Then we have our electrically charged side chains, which just means that they have a positive or negative charge. So that's going to be our carboxyl groups have lost their hydrogens, so they're negatively charged, and our amino groups have gained hydrogen, so they're going to be positively charged. So if you have an aspartic acid or glutamic acid nearby a lysine, arginine, or histidine, you'd expect them to interact and make an ionic bond because it's a negative and a positive. And so that's one way that you could either fold a protein, or if you have a charged molecule coming in, like an ion with a positive charge, you'd expect it to be attracted to and interact with the acidic, negatively charged amino acids like aspartic acid or glutamic acid. So we said the polymer of proteins is a polypeptide. The monomers are called amino acids. We abbreviate that by capital A, capital A. And when you link amino acids together, the specific type of bond is called a peptide bond. So that's like in carbohydrates, how you link with glycosidic linkages. It's the same thing, it just has a special name. And how do you make a bond? To make a bond, we do dehydration synthesis. So that's going to be here. So we take a hydroxyl group and the hydrogen group. You can see the hydroxyl is part of the carboxyl group. The hydrogen is part of the amino group. Those come together and they're going to make water. When water leaves, that means that these two bonds are empty. The electrons are no longer having a pair. So what that means is that these two, in order to fill their shells, will attach together. And then you have the peptide bond between two amino acids. So the way you can recognize where a peptide bond is, like if I gave you a chain of amino acids, you look for a nitrogen, carbon, carbon. Nitrogen, carbon, carbon. That's the sequence. So if you go nitrogen, carbon, carbon, before you get to the next nitrogen, that's where the bond is. So it'll always be between a carbon and a nitrogen. The next one between this carbon and the next nitrogen, and so forth. So you end up with the backbone bonded together with peptide bonds and then the side chains sticking off of the backbone. The amino end is called the N terminus, so that's one end of the polypeptide chain, and the carboxyl is the carboxyl group at the other end of the polypeptide chain. So it does have directionality. When you take that protein and you coil it, and possibly put it together with other polypeptides that are folded and coiled, that's what makes a protein. And you should note that the amino acid sequence is going to determine the 3D structure. So depending on what order you put the amino acids in, dictates what kind of characteristics are going to be present. So let's say a positively charged amino acid. This one's a negatively charged amino acid. This one is partially positive because it's a hydroxyl group. We have a nonpolar one here with some carbons and hydrogens. So this is going to dictate how the protein folds. So in this case, we'd expect something possibly like an ionic bond between the positive and negative, so that would fold it there. We'd expect hydrogen bonds to have formed between partial charges, and then we'd expect nonpolar groups to interact with other nonpolar groups in hydrophobic interactions. And of course, however the structure of your protein is going to determine the function. So if you have a protein like this, possibly its function is to bind a molecule that looks like that. If you have a protein that is long and spindly, has a couple different subunits, perhaps its function is some kind of structural function to elongate the cell. So here, for example, this is a ribbon structure, so you can see this is the protein part, and it's in coils, and then some unfolded parts here. I'm not following exactly right, but there's the coils. And you can see that there is this groove here. And you can see it even better in this three-dimensional structure. So here's the groove, and that's going to be the binding pocket for where this protein, which is an enzyme, binds a substrate, a molecule, and catalyzes a reaction on that molecule. So here's an example of an antibody. That's a protein. And you can see it as a very specific shape that binds, particularly, in this case, to proteins from the flu virus, which also have a particular shape. And this protein is that shape because of the order of the amino acids. For structural levels, you'll need to know the structural level name, primary, secondary, tertiary, and quaternary. The characteristics of the physical structures. So for example, primary structure is an amino acid chain with peptide bonds. Secondary structure includes alpha helices. And then you'll need to know what forces hold those structures together. Here's some examples for, this is secondary, tertiary, quaternary. That's how you write those with the number and the, looks like a degree sign.
And those are outlined in your textbook and in Mastering Biology.
I do want to show you, this is tertiary structure here, and there's all different types of bonds that can be involved. So these are all R groups sticking off the protein backbone, polypeptide backbone. And you can see there's hydrogen bonds involved, there are hydrophobic interactions involved, disulfide covalent bridges, and we even have ionic bonds. There's also van der Waals interactions occurring, and all these help hold the protein in particular shapes. So now you can see why it's so important to understand if you have a hydroxyl group, you know that that's going to have a partial charge. If you have a carbonyl group, it also has a partial charge, so they can hydrogen bond. Carbons and hydrogens are hydrophobic. They interact with other hydrophobic entities. Positive charge and a negative charge are going to form an ionic bond. So if I were to give you a sequence, you should be able to tell which structures would bind or not bind with other structures and what type of bond they'd make. So here's your overall primary to quaternary structure. So make sure you understand this figure. If you don't, let's talk about it, bring the questions to class, or come to my office hours. If you change the sequence of the amino acids, sometimes it doesn't do anything, but sometimes it makes a really big change in the protein and its functionality. So sickle cell disease is an example of that. One change in the amino acid sequence leads to a slew of physical effects in the person. So here's your primary structure. This is the normal hemoglobin. You can see here are the amino acids in their chain. So this is the polypeptide. Then there's folding that is involved in secondary, and then more folding in tertiary. Subunits are put together to make quaternary structure, and those structures here are hemoglobin. They are in red blood cells and can help carry oxygen around the bloodstream. In sickle cell hemoglobin, you have one mistake. So one amino acid is changed, and that's because the DNA has one letter change as well. So it codes for a different amino acid. Now the folding is different, and you have this hydrophobic region. Valine's hydrophobic, where glutamine was not. So the hydrophobic beta subunits actually bind together with hydrophobic interactions. This means that they form these crystals. This doesn't allow them to carry oxygen well. When oxygen's removed, it makes the red blood cells this funny sickle shape, which means they don't get through capillaries very well and can clog those up, and it's very painful, and this causes what we call sickle cell disease. So you can see a single amino acid change can change an entire protein and obliterate its functionality and cause diseases that are highly significant in the population. Proteins, the folding is strong, but it's not unbreakable. So I'm talking about weak interactions, so like your hydrogen bonds, your ionic bonds, your hydrophobic interactions. The main covalent bonds, the peptide bonds in the backbone, they stay together unless you have an enzyme that comes in and chops them apart with hydrolysis. But the weak interactions of like the helices and the beta-pleated sheets, they can be interrupted with a number of different factors: pH, salt concentration, temperature, etc. And they can cause the protein to unravel or unfold. It doesn't break the protein down, it just unfolds it. And when it doesn't have shape anymore, now it doesn't have functionality. And that unfolding we call denaturation, or the protein is denatured. That denatured protein in some cases can renature, meaning it gets folded back up again correctly. But sometimes having unfolded proteins, too many of them, is detrimental to the cell, and the cell will die. To fold proteins back up, there are specific molecules that are involved called chaperonins. I mention them because their structure is pretty interesting as far as the structure-function relationship goes. So they have, this is the real structure, but they have this hollow cylinder inside. The unfolded polypeptide goes inside the cylinder. Then there's a cap part of the protein, the subunit, that goes on top. And the cylinder is basically protected from the environment. The cylinder is hydrophilic, which allows the protein, helps it to fold. The cap comes off, and then the folded protein leaves and goes on to do its function. So you can see the structure of this protein. So it's one protein helps another protein fold, and this is very specific, a structure which allows it to perform its function. Sometimes proteins misfold. So some of the things we have problems with in humans are Alzheimer's and Parkinson's. Mad cow disease is in bovines, obviously. There are versions in a lot of different animals and humans as well. Jacobs-Creutzfeldt disease. All these are problems with protein folding. The protein tends to accumulate, particularly in the brain, starts to kill the brain cells, and then you have neurological problems. So protein folding is very important to happen correctly. And then finally, how do we discover the structures of these proteins? As you'll notice, like the chaperonin, it had the cap and the cylinder and all that, and we actually knew what it looked like. So one of the ways, the most precise way right now to do protein structure is called X-ray crystallography. So what happens is you crystallize your protein, which sometimes takes years of time to do, and so it's repeating units of that protein in a little crystal. You take that crystal to a synchrotron, and X-rays are shot at the crystal. The X-rays scatter depending on the crystal structure, which is based off the protein structure. You detect those, they form a diffraction pattern. Nowadays we can take and use computers to help analyze that along with mathematics and logic, and eventually you can hopefully figure out what the structure of your protein looks like. So for example here, the gray is your protein, And with the structure of the protein, it allowed us to understand where the DNA bound and where the RNA came out. So this is an RNA building molecule, builds it from reading the DNA. And so structure can sometimes tell us what the function of that protein might be and how it occurs. There are also other kinds of structure, protein structure determining methods like nuclear magnetic resonance, NMR spectroscopy. It's a little less detailed than X-ray crystallography, but sometimes easier. And we can also try to predict what proteins are going to fold like through bioinformatics, using computer programs to do that. There's also this interesting phenomenon going on right now with a program called Fold It. You might have heard of it. So it's basically designed like a computer game, and it's available to regular people. You can go get it online. And it's a challenge for you to take a sequence and using certain rules, a protein sequence or polypeptide sequence, using certain rules to figure out the most likely way it would fold. And whoever comes up with the most efficient and parsimonious, meaning likely folding answer, can win prizes or be put on papers. They've actually used this to actually solve some structures of proteins. So it's been pretty interesting. It's crowdsourcing with regular people to try to help advance science. So that's pretty neat if you want to go look that up.